Hello, everyone. This is chapter 1 of your microbiology text, An Invisible World. The main student learning objectives is to describe how our ancestors improved food with the use of invisible microbes. So fermentation is as old as man himself. So we'll see how our ancestors preserved food. We'll also study the causes of sickness and disease and how it was explained in ancient times before the invention of the microscope. And we'll also talk about some of the most historic events in microbiology. And this is an overview of what you'll be studying this semester. We'll talk about some of the important microbes like bacteria and protist, fungi and virus. So here's a nice animation of how there's a lot of bacteria here and the white blood cells are able to engulf it. It's your immune system of your body. So microbes can be seen only with the aid of microscope. So it's ubiquitous. That means it's found everywhere on Earth, in the air, in the water, in plants, in animals, in the dirt. Without microbes, life as we know it can't exist. Everything in the world evolved from a microbial ancestor. Microbiology is the study of microbes. It is found everywhere. So the word for that is ubiquitous. And you need a microscope to see it. That's why it's called microorganism. Some that we will study about, like worms and some fungi, are visible to the human eyes. Most of them are harmless. There are some documented very harmful ones. Without microbes, life can't exist. Life as we know it evolved from microbial ancestors. There are microbes that is maintaining the oxygen content of the air, like algae and cyanobacteria. So the oxygen content that's very important for survival is maintained by microbes. They keep us healthy. We've heard of the microbiome of your GI tract that will help in digestion and absorption of nutrients. They are sources of new drugs, like how we get antibiotics, like penicillin. Helps us to digest food. There are microbes that can clean up hazardous waste in the environment. You probably heard of oil-eating bacteria, so they play a huge role in bioremediation. They also support and protect agriculture. So there's nitrogen-fixing bacteria in the nodules of certain plants. It has a symbiotic association with plants, and they provide nitrogen for the plants. So the role of microbes is tremendous, and they are very, very vital for life on Earth. So our ancestors, this is like, there's evidence for fermented foods, even 5,000 years ago. The Egyptians knew it, the Incas, the Mayas. So when we study these old civilizations, there are records to show that they enjoyed bread and beer and wine, fermented foods. So fermentation is not only for preserving food, but it also enhances the nutritional value of food. For example, the yeast, the microbe yeast, can break down glucose to form ethanol and carbon dioxide. Ethanol is alcohol. So good examples would be beer making and bread making. So there's evidence of this happening on Earth 7,000 years before Christ. The fermented beverages by yeast, Saccharomyces cerevisiae, or bacteria can be used. The bacteria called lactobacillus can be used to break down milk to form yogurt. So lactic acid is responsible for the sour taste of kimchi, sauerkraut. All these are fermented foods, natto, tempeh, kombucha, and this kefir, and this all these fermented foods. So we have yeast fermentation to produce alcohol, and we have bacterial fermentation to produce lactic acid. So the science of fermentation is called zymology. So what are our early notions of disease, contagion, and containment? Again, there's endless proof to show that our ancestors did have some clues about what caused disease. There's evidence in the Bible about how people with leprosy were quarantined. So they knew there was something causing the disease, and they knew that quarantine helped to manage the spread of the disease. And here this picture is Otzi, the Iceman. That is about 500 years ago. So this mummy was infected with a parasite and a bacterial infection, and it is known that he, like a lot of forensics on Otzi the Iceman, shows that he was suffering from Lyme's disease, and he was self-treating the disease with some fungal elements and herbs. And here's a picture of a blueprint of a Greek city. So they have extensive aqueducts for fresh water and sanitation and hygiene practices. So they attributed disease to bad air. It's called mal air. They developed hygiene practices. They have very good sanitation infrastructures to deal with sewage. So they kept the water supply separate from the sewage system, because they knew that these can be sources of waterborne illness. So let's talk about the history of microbiology. Again, it has very, very beginnings from thousands of years ago. We'll talk about the major ones. So about 400 years before Christ, there's Hippocrates. He's known to be the father of Western medicine, because he postulated that disease that we see in humans had a cause. It was not caused by sin or curses and so forth. Here's Thucydides, father of scientific history observations. He made observations about the concept of immunity. There's another important person called Yarrow, and then he proposed that diseases were caused by some elements that we cannot see. So it's not like they were clueless. History shows that they had some idea of the contagion. Anton Leeuwenhoek was the first to develop the simple microscope. So he was the first to view microbes. So this is early 16th century. And then it was only like after 1850s, a lot of microbiology discoveries and inventions were made. So it's called as the golden age of microbiology. Early 18th century, 1820s, Louis Pasteur demonstrated that fermentation is caused by microbes. And then pasteurization, named after him, is the process of killing the harmful microbes in food. They would, like, for example, milk, when they heat it, you can kill most of the bacteria to make it safe. He also developed some vaccines, especially rabies vaccine. Robert Koch identified specific microbes for diseases. So specific microbes for specific disease was first postulated by Robert Koch. Very important. So how are we classifying the microbes? What is taxonomy? So taxonomy is classifying organisms. So the science of naming organisms, like you identify the microbe, describe it, and classify it. So that is what taxonomy is about. And the father of taxonomy is Carl Linnaeus, 17th century. So he divided, see here, Carl Linnaeus, he developed a way to categorize. For him, at that time, before the discovery of the microscope, it was plants, animals, and like one more unknown kingdom here. And then in the 18th century, we have Ernest Haeckel. He proposed the two kingdoms of Protista and Monera. So that was added. The science of studying the evolutionary relationship between organisms is called phylogenetics. So what you're seeing here is the phylogenetic tree. So here the base would be the ancestor, and then that, because of selection pressures, has formed these new species. So this is called as the phylogenetic tree. So you can trace back relatedness of organisms. Another important person is Robert Whittaker. So he added the fifth kingdom, the fungal kingdom, fungi, to the four. So throughout history, we can see how scientists added on to the understanding of taxonomy. So he combined all the five kingdoms, and they were so with this Monera, Protista, plants, animals, and fungi. So this was the standard. This was followed for many, many years. And now genetics and gene sequencing came into play. So Carl Woese created the genetics-based tree of life. So he would sequence ribosomal RNA, because this is the sequence that can get mutated first. So if you want to study genetic relatedness among organisms, this is an excellent tool. So what happened was, after molecular biology came into play, the tree of life was rewritten. So he created a tree with the addition of the domain to the taxonomic structure. So here's the phylogenetic tree that is most widely accepted now. So we have bacteria, archaea, and eukaryotes. Bacteria and archaea are prokaryotes. So this is before a well-defined nucleus evolved. So they do not have a membrane-bound nucleus or most of the membrane-bound organelles. Whereas eukaryotes are more sophisticated, and this is where you see the fungi, the protists, plants, and animals, and those kingdoms here. So the five kingdoms of bacteria, archaea, plant, animal, protist, and fungi. And there are three domains. Archaea and bacteria will contain prokaryotes, and eukarya contains eukaryotes. So this is modern taxonomy. You have to know this. So now how are we going to name microbes? There's literally thousands and thousands and thousands of microbes. They outnumber us. They are more diverse. And like new microbes are discovered constantly. So how are we going to classify them and name them? So we are going to use the same binomial nomenclature proposed by Carl Linnaeus, father of taxonomy. So every living organism, like for us, we are Homo sapiens. We have a genus name and a species name. So very similarly, all the microbes are also named by the two-word system. It has a genus name and a species name. So both the names will be italicized. The genus name is always capital, and they have Latin and Greek roots. Sometimes they're named after the geographic location, sometimes it's named after the scientist. For example, Escherichia coli was named after Theodore Escherick, who discovered it. Coli stands for colon, where it infects or where it thrives. Staphylococcus aureus. Coccus means like bunches of circles or grapes like. Aureus means like a golden color. The colony is golden color. A series of manuals that described and developed as these microbes were discovered is called a Bergey's manual, developed by microbiologists to identify and classify microbes. So let's talk about the nature of microbes. So microbes, micro means very, very small. It's a very small entity. You can't see it. So for example, the human eye can see like 1 millimeter, or maybe like some people even a tenth of a millimeter. But for anything smaller than that, in micrometers, 1 micrometer or 10 micrometer, you need a microscope. So we'll be using a compound light microscope to see bacteria in the lab. So here you can see like a plant cell and animal cell is between 10 to 100 microns. A bacteria is only 1 micron. Whereas if you go on to virus, like a polio virus here, it's only like 10 to 15 meters or nanometers. And then we have lipids and atoms and proteins. For anything smaller than a micron, you would need an electron microscope for seeing it. Most microorganisms are unicellular. However, there are some that are multicellular. Most of them, you need a microscope to see it. However, some larger ones like mushrooms that come under microbiology, you can see it. Some microbes are acellular and not living. That a good example is a virus. And microorganisms are found in all the 3 domains of life. But virus is not placed in any domain because it is non-living. So you need a microscope to study most microorganisms. So coming to bacteria, so they inhabit every environment on Earth. Most of them are harmless, but some of them are pathogens and they can cause disease. They are mostly unicellular. They are unicellular, and their cell wall, they contain peptidoglycan. There are photosynthetic forms and non-photosynthetic forms, and they are described by the shape. A circle is a coccus bacteria. A rod one is a bacillus bacteria. Vibrio is curved. Spirillum is spiral shape. A spirochete is a long, slender, spiral-shaped bacteria. It's like a screw, this one. And they are capable of movement. Coccobacillus is a combination of both a coccus and bacillus. So we identify bacteria and classify them depending on their shape, coccus, bacillus, and so forth. And another thing to remember is that they have a very unique cell wall containing peptidoglycan. It's a type of carbohydrate. So let's look at some of the microbes. Here's archaea. Archaea live in very harsh environments like a geyser or a muddling, like a bubbling mud pot here, or like a hot water geyser here. So they are prokaryotic and they are unicellular. They do not have peptidoglycan. That's why they are in a different domain called archaea. However, they have special lipids that are branched, which are not found in bacteria. They also differ by having different types of ribosomes and gene sequences compared to bacteria. And the unique thing about them is that they can live in like harsh environments, like high salinity, alkalinity, like acidic environments, and like boiling environments, extremely hot environments. So that's, see, methanogens can like grow in methane, like, so that's what differentiates them from bacteria. Protist, kingdom Protista is huge and it's very interesting. Again, it's the taxonomy is very detailed. So this can be unicellular or it can be multicellular. For example, amoeba that you've seen in labs before, um, paramecium, they can be unicellular, and there's also multicellular algae like this. They can either be heterotrophic or autotrophic. So sometimes if they don't have chlorophyll, they have to eat food to get the energy. Sometimes they can make their own energy. So it's broadly classified like animal-like protist or plant-like protist or fungus-like protist. A good example for animal-like protist, they're called protozoans, is amoeba. Plant-like protist would be algae. Fungus-like protist would be slime modes. But it does not like have chitin in their cell walls like how fungus does. Some protists that cause disease, for example, malaria is caused by Plasmodium falciparum, sleeping sickness by Trypanosoma brucei, and diarrhea can be caused by Giardia lamblia. One of the most important aspects of protist is that half of the oxygen in the atmosphere comes from the oxygen made by autotrophic protist. Like they are unicellular algae. You probably must have heard of phytoplanktons, diatoms, dinoflagellates. So not only are they maintaining the oxygen content in the atmosphere, but they are also in the bottom of the food web. So the thing is, they play a huge role in the ecology. Fungi is very important. So here they are eukaryotic. Protist also coming under eukaryotic. They are heterotrophic microorganisms. They are both, there are some that's unicellular like yeast, and there are ones that are multicellular like mushrooms. The important aspect to remember here is the cell walls are made up of chitin. One yeast that causes disease in humans is Candida albicans, that causes like a vaginal yeast infection or oral thrush. One economic advantage is some of the molds can be used for making medicines or pharmaceuticals. For example, the penicillin mold can make penicillin, which is a widely used antibiotic. And fungi, the different types of fungi, here's like a mushroom and all these, here's a bread mold. So these fungi, they play a huge role in recycling of nutrients in the ecosystem. So they cannot make their own food. They are decomposers in the ecosystem. Because they are decomposing the dead material, they are able to recycle nutrients in the environment. So they are very important. And of course, yeast is coming under fungi. And you know how important yeast is in the production of beer and wine and all the fermented products and bread. Helminth is referring to worms. They are eukaryotic microorganisms. They are multicellular parasitic worms. Most of them you can see like macroscopic. However, the eggs and the larval forms are microscopic. And here's Taenia saginata, the beef tapeworm. Eggs are microscopic, and adult worms are like very long, up to like 4 to 10 meters, taking up residence. They suck up all the nourishment of the host, and they are very clever. They can camouflage and escape the immune system. And here's a man with a lymphedema. So there are like worms that cause, maybe this is the one Draculanucus, that causes lymphedema. So that's why sanitation and cleanliness is very important, because the spores are many a time are found in dirt. And like when children, like when adults, like if you don't wash your hands, it can get into the body and start living there. Viruses are super important. And as we think about the current pandemic, it touched every aspect of life. Thousands of people dead. So it is not living. So this doesn't follow any of the definition of the rules of life. It is acellular, non-living entity. So if you take the genome of the virus, it does just have either DNA or RNA as the nucleic acid, and it has a protection of proteins around it. It cannot perform any of the activities that we associate with life, like respiration or any kind of metabolism or reproduction or digestion, none of that. It can only live inside a living host. So it's called obligate. So it will enter the host, take over the host metabolic machinery, and then it will make many copies of it, and then it will lyse and come out. The study of virus is called virology. Again, viruses outnumber anything else on Earth. It's very small. It is found in every ecosystem. It's the most abundant type of biological entity. It can cause very severe disease. So microbiology as a field. So if you study microbiology, the study of bacteria is called bacteriology. So there are people who specialize just in bacteria. You know, they are experts. When you fall sick, when you send the blood to the lab, you know, they will grow the bacteria and they will identify the bacteria, and they'll also come up with the correct treatment to kill that specific bacteria. So that comes under bacteriology. Mycology is study of fungus. Protozoology is protozoans. Phycology is algae. Parasitology is parasites like helmets. Immunology is the study of the immune system, and virology is the study of virus. Nematology is the study of nematodes. These are like microscopic worm-like organisms. So this chapter, summarizing this chapter, we just had an overview of what we'll be covering this semester. Thank you.